So vaccines in general are things that are made from small amounts of weak or dead germs, things like bacteria and viruses, toxins, that sort of thing. And we use those in order to prevent disease. So an investigational vaccine is one that has been approved by the United States Food and Drug Administration, or the FDA, for use in a clinical trial in humans. And it's not approved at this point to be used out into the general public.
Gotcha. So what are some of the testing procedures that are gone through for each vaccine before it enters a clinical trial?
So there are a couple of things that go on before we introduce it into humans. The first part really is an exploratory part. It's done by researchers, usually in academic institutions, and using their expertise as researchers with that disease and with vaccines, then they kind of come together and think about ways that they can come up with a vaccine to prevent a disease. So that's an exploratory stage. It usually takes about 2 to 4 years to do that. And then from there, they move into the preclinical testing. And that's really where we think about test tubes and when we talk about animal testing. So they'll start with test tubes, they'll put it into a test tube and see if that vaccine will prevent something happening in that test tube. And then from there, they move on to animals. And really at that point, we're looking for safety, making sure that it seems to be safe, and then also that it's producing an immune response. So when I say immune response, I mean like your body's reaction to having that in there in a protective way. And then other things that we'll look for at that point is the dose. What kind of dose should we be giving? How should we be giving it? Is it given up the nose? Is it given as an injection under the skin? That sort of thing. And then also perhaps how many doses do we need to give? 1, 2, 3 doses, that sort of thing. And then they'll use that to adapt it into how should we move this forward into humans.